now I'm trying to gauge what sort of tools do we use to gauge whether a worldview is correct philosophically or scientifically. So it depends.
For me personally, I would, and I give everybody my word, I would actually accept evolution as in common descent being true if I can only figure out if the mechanism can actually do what's claimed, down the long run. But I've talked to a lot of people over that, and just, talking about predictions, it's just not very convincing, because, like I said, when I look at it, the same predictions can be made that a creationist can make, and if we're looking at things according to our presuppositions, that seems like that's really the only thing that's happening. So I want to know. I want to know if small changes lead to the big changes.
Yeah, that's there are two. True, if you don't mind, there are two things I'd like to say about that, and one is that I have heard you say before about how that what you really need is, what was it, proof of the mechanism, or what would you say? Can you just repeat that phrase again?
I want to know if the mechanism can do what's claimed, in the long run as a common descent.
That's kind of, in terms of scientific inquiry, that's kind of ass backwards through the cat door, because the first thing you need to do is to demonstrate that the phenomenon takes place. So for example, if you claimed to have psychic abilities so that you could know what the winning lottery numbers were every week, obviously we'd all be skeptical of that claim, and we might ask you something, well, how does that work? And you might give us a theoretical account that includes a mechanistic explanation for how you're able to do this, and it may involve making, hats out of tinfoil, or who knows what your theoretical account is. And if we just say, well, there's nothing to see here because I don't believe his theoretical account, and just walk away from it, what we've done is we've missed the first important step, and that is to confirm that the phenomenon actually takes place. And if it turns out that you go ahead and make a prediction that week, and you get 1 out of 6 numbers right, or 2 out of 6 numbers correct, and you say, there, see, it's not a perfect system, but I got a couple right. Well, then what we have to do is compare it to the base rate, like what our expectations were under the theory that you don't have any special powers to predict the lottery numbers.
Right. Yeah, but what if you predicted all the numbers?
I think the problem is that.
Then what that shows.
Let me respond briefly, please, because I know that other people are going to want to jump in. It seems like, so I just want to respond really quick. I think that the only way that we can see if it can happen, or see if it happens, is by knowing if the mechanism can actually do it, right? Because we can't say that it has happened before without knowing if something can make it happen, right? So I mean. That's the reason why it's just not a satisfactory answer to compare the homologies or the similarities of species that you're looking at in the fossil record. Because that's declaring evolution was behind it is not the same thing as saying that, well, the small changes were actually behind those big changes.
No, and that's what I'm saying. That's exactly that. That would be science backwards. I might even be generous to call it science backwards, because let me extend the example of the lottery ticket thing. Okay, so what happens, as Stephen asked, what happens if I say, well, I don't really believe your story about, or like maybe you say, let's come up with something really crazy. Let's say that the reason you say that time traveling aliens. I don't believe in time travel. I don't believe in aliens. So for you to tell me that you're able to know what the lottery ticket numbers, the winning lottery numbers are, because of your time travel aliens, I don't believe the mechanism. I would really struggle to get some proof from you that these time travel aliens really exist. But therefore, I'm going to dismiss your claim that the truth of the matter is you're able to predict these numbers. So, okay, what happens then? I'm skeptical of your proposed mechanism, but should I dismiss the factual assertion that you have the ability to predict the lottery numbers? No, we should test that. You always test the state of affairs first.
Right, but then how do we test it? We test it.
Okay, well, in this case, let me do the lottery number thing. Let me do the lottery number. So let's say you do come up with, you get all 6 numbers right. And I'm like, well, okay, that's amazing. That's 1 in 14 million. And so, well, no, hang on. I'll do it again for next week's draw. Well, now it's 1 in 14 million squared. So now I'm pretty seriously convinced that some phenomenon is taking place. I don't have to accept your theoretical account. Now what I have to do is say, well, what other predictions would emerge from that particular theoretical account? Now, there may be another theoretical account, and that is that you have a cousin that works for the Lottery Commission, and the whole thing is a fraud, by the way, and your cousin's been feeding you the numbers. Right? Now, that's also a bit of a stretch, not as big of a stretch as time travel aliens, but that's another possibility. Under that theoretical account, it means that at some point you're going to take a secret phone call or get a piece of email or something. So I set up surveillance, and I find out, yep, sure enough, Thursday afternoon, you get an email from your cousin that has the winning numbers.
Now,
right?
question: What if no one messaged him the numbers, or he doesn't have a family member or a friend or anybody he doesn't know? He's not affiliated with anyone that works in the organization.
Okay, well, who cares? Who cares? When it's your turn to come up with a hypothetical, you go ahead and you run with the hypothetical. Let me bring mine in for landing. Okay. The point is that if the fact in this case that my example, the state of affairs is you do have a special power to predict what the lottery numbers are. There are two different theoretical accounts for how that might be, and just because I dismiss your first theoretical account as being something that I can't get my head around, or for whatever reason I dismiss it because it's inconsistent with other things I believe or whatever else, means that I'm going to dismiss the phenomenon itself. It means I missed the opportunity to engage in the investigation, right? So obviously we confirm that the phenomenon exists. Then we start to look and we surveil. And I look and well, you get an email from your cousin, or let's say you don't get an email from your cousin, but instead one night the sky goes green and clouds come in and there's a UFO comes down and one of them hands you a little ticket with the numbers on it, right? Right. So you don't investigate. You cannot. My point here is you cannot dismiss the phenomenon because of your resistance to believe the proposed mechanism.
But the problem is that that doesn't, the phenomena, I mean, what phenomena can we connect with evolution? I mean, I don't see where your point is leading. So, I mean, what are the phenomena that we can't dismiss?
Hold on, so let me...
The fundamental point of disagreement, whether or not humans and apes have a common ancestor, or humans are a type of ape. That's...
Well, just, I think that's
the
different thing, though. Isn't that just like a claim? I mean, because how do we know that we had a common ancestor? I mean, because we can't just look at some organism a long, long time ago and then just, like, declare it, well, this looks more similar to us, so therefore, you know, this is our common ancestor. I mean, that's what I'm saying. It's just like
that's not how it's
declaring it. We're just declaring things, and it seems like this to me. It seems like this is what it is. We're using really mainly our imagination to connect dots. So if I come in with a presupposition, I can picture it in my head, like, you know, progression. But that doesn't tell me as if, you know, evolution actually did it.
Sure, and that's not how it happens. That is not at all the process for how the conclusion was arrived at.
But isn't that the exact same analogy? That's the analogy you were giving me, right?
No, it's first.
It pretty much comes down to that.
To ask you a question, Hogtie.
No, that's the way religion approaches this subject. That's not the way science.
Look, that's not the way that religion approaches the subject. We're not talking about religion. We're talking about evolution. And I understand how science works. That's the reason why I agree with him. That's not the way that science works. But this is the way it seems like evolution works, and this is the reason why I can't accept it. I mean, with other theories, like Einstein's theory of relativity,
predictions. That's how evolution works, and also is falsifiable.
Eddie, you're always bringing things like that up. What testable predictions can you bring up?
Yeah, the evolutionary thing.
You better let him answer.
Okay, yeah. I just want to ask a question afterwards.
Yeah, Antonio, you can go next, but I just want to hear Eddie because he seems to be making shots, so maybe he's got something he'd like to bring to the table.
Yeah. No, I mean, Darwinian evolution has made several predictions. I mean, just one was the fact that there are two kinds of whales. There's whales with teeth, and there's whales with balines. It was predicted long ago that we would find a transitional whale that, once it existed, had both teeth and baleen. And that was found. And that was something that could not have been predicted by creation, by any kind of creation science, or it wouldn't make sense. It only makes sense.
Well, what whales? Because we already know that certain whales have teeth. So I mean, I don't know what you're talking about. You got to be a little bit more specific here.
Evolutionary theory predicts that there must be if whales with teeth and balines had a common ancestor, there must be a whale that has teeth and baleen. Even though there's none that exists now, it predicted that we would find it in the fossil record, and we did.
Okay, but that's not really a prediction. I mean, how is that a prediction for it? And first of all, who cares? I mean, they have teeth. That doesn't mean anything, man.
No, first off, it is definitely a prediction, because before that prediction was made, no one had made the observation of a whale with mixed denture. And then after the prediction was made, they found fossils of whales with mixed denture. And at the depth and with the other same morphological features that were of a more generalized whale type as opposed to modern specialized whales. So it is exactly the definition of what a prediction is.
Well, no. First of all, I would like to see an article for that. And the second thing, based on what you guys are telling me, that is not necessarily a prediction for evolution as in common descent. I mean, just because you can find something else that you can put in there as an intermediate doesn't necessarily mean...
Let's bring the goalposts back to where they were.
Thank you very much.
Over there.
Eddie, I can't hear him. Eddie, when you over-talk him like that, I can't hear him. Can you go again, Hogtie?
Oh, I was going to say, let's put the goalposts back where they were. You can shift them later.
Oh, there's no goalposts. There's no shifting the goalposts.
It is because...
It's coming back to exactly what I'm saying. Remember what I was saying earlier about the apes? Let's just say that we can find an ape a long time ago that looks very similar to us, right? Let's just say that we can find an ape a long time ago. Just because it looks similar, that doesn't therefore mean that we can declare that it was our common ancestor. And that's exactly what you're doing right here.
And nobody does that. Nope.
You're doing the same thing right here with the whale bones, with the whale teeth, and you're declaring that because this one looks similar, therefore it came from it. This was already dealt with like 20 minutes ago.
No.
So what's the difference?
Isn't that it? Yeah, it is.
I'll tell you. This is actually one of the. We haven't been doing this conversation for a while. This is one of the things that we get from one of those creationists or ID proponents or whatever else. Is the presentation that the way that conclusions are arrived at for people who accept evolution. The idea here is that they look at this one thing and they say, oh, well, obviously man came from monkeys. And I hear this all the time where people say, oh, well, you look at this similarity and then you conclude evolution's true, and there's no way that proves that. Or you look at genetic similarity and then you say, well, the evolution must be true. Well, that doesn't prove that. And you notice every time, and again, it's hard to tell sometimes like whether or not when people interrupt or speak at the same time that you can hear it. But true, every time that you said something like, well, that doesn't prove evolution, I said, while you were saying that, I go, yeah, you're right, it doesn't. I always agree whenever we're talking about a single observation. So let's go back to the whales, right? So with the mixed denture, the baleen versus conical teeth, there's no whale alive today that has both. They're very specialized. They have conical teeth, which are really good for eating fish, and then there's the baleens that are really good for straining these little critters. Yeah, and there's no creature that does both.
Plankton, I believe it is.
Plankton, yeah.
I say critters because I'm not a biologist. Nor have I ever claimed to have a PhD in biology. I don't want that out there that I've ever said I have a PhD in biology. Never made that claim.
So krill coming from Steve thing out, really?
I saw that.
I don't know why he attached that to me. That was really weird the way that he did that.
It wasn't you. I know it wasn't you.
No, it was.
As soon as he said it, as soon as the other guy said it, you went, "Oh, really?" Because you'd never heard it before, and that was fair. I just.
Yeah, well, I'm saying, I mean, I wouldn't be surprised to find anything, more of the same thing, more of the same variety. 99% of the species are dead. But, I mean, we can't just find a species that looks similar and just declare, well,
but we don't. That's not what
I know you're saying it's a prediction. I get what you're trying to say.
Right, it's a prediction.
You're saying that's a prediction, but the prediction is just it's off because is it really a prediction? I mean, we're going to expect to find more if 99% of the species are gone and extinct. I mean, we see the same thing.
That is a prediction. Because it's a prediction that logically follows from the theory. So we have these things that are.
But that's the problem. I'm not finished with what I'm trying to say, though.
Okay, sorry.
That's the problem. That's the problem, though, because the prediction doesn't necessarily logically follow from the theory. It doesn't logically follow that, therefore, common descent was behind it. I mean, how do we know that common descent was behind it? I mean, I could just sit over here and say that all three species were therefore created at once. I mean, who's right and who's wrong? The one that says, well, I'm finding an intermediate here, therefore, it doesn't follow.
Okay, that's the goalpost shift. What you're doing, because what you asked him for was a prediction from the theory. What that means is that you're saying, if the theory, and specifically when I'm talking about evolution, I'm talking about common ancestry specifically, if the view that all extant whales actually came from a more generalized, less specialized common ancestor, then give us an example of a prediction. If that's true, show us an observation that hasn't been made, and then they made this prediction, and then now that observation was made. Give us an example of that. So he gives you an example of, well, look, we've got whales that either have conical teeth or they have balines, because they have totally different, feeding styles. But if they really came from a common ancestor, necessarily there must have been something out there with more generalized teeth. They weren't specifically conical, they weren't specifically baleen. There must have been, or mixed denture or something like that. So that's a specific prediction. And then after they made the prediction, they found an ancient animal that had that. Does that prove that evolution is true? No, there may be other possible accounts for it. But you asked him for a prediction, and he was able to handily provide you with one. Then you turned around and said, yeah, but that doesn't prove evolution. If you asked him to use whale denture as an example to prove evolution true, well, then he failed, but that's not what you asked him. You asked him for a prediction, and he provided one quite handily.
Mm-hmm.
Well, that's the point, though. The point is that I'm asking for common descent. That's the whole entire point of this discussion. Like, I want to. He's saying that, well, this is what, and correct me if I'm wrong, Eddie, but you were using this as an example to say, well, this is the difference between science and religion, right?
No, yes, well, sort of. My point was, and you made a point earlier on about how religion is not open to change or open to, I forget how you put it. I tried to interrupt, but I wasn't successful. But you mentioned something about this, about how, anyways, about how evolutionary scientists are not open to change. And, what I was trying to demonstrate is that evolution meets the criteria of the solid theory because it has these predictive qualities. Not only that, but it's falsifiable. You can actually falsify these predictions that evolution makes. You can find that's false.
That's what you were saying. Yeah, no, that's what you were saying, and that was the point. That's the problem. I mean, we would make the same types of well, maybe not this prediction. I'd have to admit, maybe this specific type of prediction, but we would predict. I mean, some of the same things that the evolutionists would predict. And this prediction right here, I mean, I want to know, how does this follow, therefore, an evolutionary model, as in common descent? Well, because that's what I'm interested in knowing about.
Hold on. Okay.
Well, I haven't spoken for a while.
No, go ahead. Go ahead.
I did put on the side.
Well, Antonio wanted to actually speak right here. Antonio actually wanted to speak right here, but yeah. I hope that we're getting that, because that's what I want to know. I mean, how does?
I'll be very quick.
Okay, well, I'm talking already, and I should have let Antonio speak. So go ahead, Antonio.
Okay, I just had a question for Hogtie. There's a mechanism for evolution that's said to be, already testable, that, something that we can practically access now. When you brought up your lottery example, that particular example seems to be an example that's more practically testable and accessible within a smaller range of a window. Now, when we're talking about evolution, that seems to be like smaller changes over time, so the window seems to be a lot bigger and a lot more difficult and a lot less practical to access. I just wanted to get your take on that.
Yeah, I think you're right, and I think I did what a lot of people do when they talk about evolution. They want to sort of compare it to other fields of science. Remember, I opened with the example of a paper airplane, where you and I could sit around one afternoon and do literally hundreds of observations with paper airplanes and make little changes that we control ourselves, and we don't have to go to an ethics review board to decide how we're going to change this airplane. And of course, we're not dealing with millions of years, so in evolution definitely suffers that. Any sort of discussion about things that have happened over long time scales, like geology or cosmology, like it's very different that way. So in terms of the testable mechanisms, now the thing is, you can't then. It never helps any scientific theory or any scientific investigation to say, well, we don't know this and we can't know this. Well, what, therefore we're right? As soon as somebody says we don't know this or we will not ever be able to determine this, that doesn't help any theoretical account. That ends up being an information void. And I think that's one of the situations when it comes to discussions of origins, is that no, we of course can't know all these things. But let me just really specifically on your point about the mechanisms being testable. They are testable in one way, although I'll kind of have to step on a point that I heard Gio make earlier, when he was drawing a distinction between natural selection and artificial selection. I really wish that the term natural selection hadn't caught on. I mean, it was right in the title of the subtitle, I guess, of Darwin's very famous book. And it sort of described selection that was being done by nature. But there's nothing about natural selection that is necessary for evolution. What is necessary is differential breeding success, so that you have a population of critters, and some of those critters are successful in reproducing and having viable, reproducing offspring, and that there's some criterion for determining which ones are going to be successful. And that's all that matters, is that some are successful and some are not. And if it turns out, well, the one with the longer beaks tend to be the ones that are successful, well, then that's differential breeding success. That's selection. There's nothing, it's not required that it's natural selection. It could be, cattle owner selection, or laboratory scientist selection, or it could be deity selection. There could be a god that's picking which animals survive and which ones don't. That's not the point. The mechanism is differential breeding success. The mechanism isn't properly natural selection, and then therefore other methods of selection that aren't, from granola Mother Nature somehow don't count as evolution. So we can let's speed up my answer here, is that we can test those things by looking at things that have faster generations and doing selection, observing selection over things like the myelinase bacteria and whatnot, or doing those experiments in a laboratory. But I'll have to concede this then, that only works when you want to make speculation about how bacteria evolve, or Drosophila fruit flies.
And Hogtie,
the problem is,
I have some I want to throw out here.
But it doesn't help us understand how hippopotamus or elephants or human beings evolve, because those
Right, just really quick, guys. Let me respond really quick. I mean, I think this is like the point that we got off of earlier. That's testability. We can't really test to see if any of those small changes are going to lead to the big change. So, I mean, I think that's what the main point is. We can see, I mean, everybody here would agree that we're going to see variations happen. I mean, even what he brought up with the myelinase, the ability to digest myelinase. I mean, yeah, but it had to lose what? Three different functions, I believe, so we can get a function that might be conceived as something new. Does that therefore follow evolution, as in common descent? I mean, that would actually fit a young Earth creationist model, because what they're expecting is that things are degrading, things are falling apart over time, so it's losing three functions, really. So I mean, the main point is this. The main point being is that any amount of small incremental changes, any ability to to get larger beaks or smaller beaks that we would expect in different environments or whatever, that doesn't therefore mean that those small changes are going to lead to one of these big or greater changes that we're seeing in the different phylum. So I mean, I think that's the point. That can't be tested. All we're really doing is we're looking at the fossil record, and we're interpreting that based on this presupposition of evolution.
No, actually, that's
okay. Hold on. No.
I cut off Eddie, so let Eddie speak.
MMA needs to talk.
No. I've been quite a few times.
Okay, MMA. Go ahead.
Yeah.
MMA.
So, just so everyone knows, I wasn't presented to everyone, but just for the general public who watches this in the future and currently, I provided both in the side chat and was screen sharing quite a number of papers dealing with the phylogenetic tree and two recent renditions of it, one being only published a few weeks ago, and they have to go through a genomic analysis. I then shared another paper earlier on explaining how we use genomic analysis to show common ancestry and so forth. And so it's quite clear that the common ancestry, speciation, and all this has been established within science. It's obviously a blunt point to put it, but it isn't up for argumentation within the scientific community.
I disagree 100%. 100%, I disagree with that, because a lot of these species where they say that that a speciation event has happened, they're only saying that because thus far they haven't been seeing interbreeding, but that does not necessarily mean that they will not be found interbreeding in the future, and in some cases they end up being found interbreeding. So just because you find a species that will not interbreed because they're maybe separated or they just haven't been seeing interbreeding yet, that doesn't necessarily mean that a speciation event has actually occurred.
Well,
So I mean, that's not true at all. That's 100%
it's
not
not
true.
seeing them, have sex. It's about their genetics. They're incompatible. It's a genetic barrier that they are just isolated genetically. They cannot interbreed when you have speciation to an extent. You're not going to have that. And it's not about you don't see them, mating. It's
Okay,
just
so you
that
said you
it's
screen
genetically
shared,
not going to
right?
happen.
You said you screen shared. You put the links on the side, right? So which link is this?
The ones I provided on the side chat were on the two phylogenetic trees. One was from last year. The other one was
Yeah,
published a few
they don't say
weeks
the name.
ago.
So which one is it? Can you just put it again here on the side chat? I have a question to ask before we start reading it too. Before we start reading it, I have a question to ask. These articles, are they about living organisms that we've tested and that we've observed their changes happen?
Yeah, this is doing genetic analysis. Yes. So this one I was again I'll post it once more. This is the one I posted a few weeks ago. Titled "A New View of the Tree of Life," published in Nature Microbiology. And then the other one I provided was from the National Academy of Sciences, Synthesis of Phylogeny and Taxonomy into a Comprehensive Tree of Life. And I'll provide that one.
Okay, so this is the one. I'm going to screen share it, right? I'm going to screen share it. So this first link that you gave me, this has to do with the phylogeny situation, right? Or the
phylogeny tree, yes.
Yeah, so so they can't interbreed anymore. This is something that we've observed from species currently, right?
Well, this isn't going into speciation. It highlights speciation, but it's not giving you examples of it. It's just talking about the tree of life, doing this genomic analysis on a whole bunch of different.
Okay, so which one are you talking about? It's showing about speciation events happening now to where they cannot no longer.
Okay, well, it's not speciation. In that hangout me and you had before, you were reading them back out to me, and you didn't like those, but I will provide them again.
No. You've never given me any links over this before.
No. You were in that hangout, remember, and you read them back out to me, and then you kicked me eventually. That hangout.
No, that didn't have anything to do with this. The only articles that you've ever given me had to do with cancer. You were giving me the
No, and then I provided the other ones.
And the first one that you gave me had
come on,
don't start this. If you got the link.
I did from Biomed Central. I'll pull them up again. BMC. Those ones had to do with ring speciation.
I'm asking for the link.
Okay,
a lot
you
of
read
times
these back out to me.
Stuff like this, the article doesn't say anything that you're saying, so I would like the link,
Well,
please.
no, you read them back out to me. And
so is this? No, I didn't read this out. I don't think so. Let's check it out. So you're saying that this link that you just gave me just now has to do with a speciation event. That's been proven. That has to do with organisms that we've observed now.
Those two are the papers that I provided that you were reading back out to me.
Okay, so let's read this. Let's check it out. I'll screen share it so that everybody can see it. Okay, can you guys see it?
Can't see it. No, I can't see it. Oh, now we can.
All right. Okay, so it says predictors for let me see. I'm going to turn my screen. Reproductive isolation in ring species complex following genetic and ecological divergence. Reproductive isolation, RI, is widely accepted as the important checkpoint to the diversification process, since it defines irreversible evolutionary trajectories. Much less consensus exists about the process that might drive RI. Here we employ a formal quantitative analysis of the genetic interactions at several stages of divergence within the ring species complex. To assess the relative contribution of genetic and ecological divergence for the development of RI. Results by the argumenting previous genetic data sets.
Could I interrupt for a second? I mean, this is a really long article. I mean, it's really going to be
he can just read the
conversation to read this entire article.
He can just read the results and the conclusions. It goes into more depth, of course, but just read those results and conclusions.
Okay, so the results and conclusions. Okay, that's where I'm at right here. By augmenting previous genetic data sets and adding new ecological data, we quantify levels of genetic and ecological divergence between populations and test how they correlate with restrictions of genetic admixture upon secondary contact. Our results indicate that the isolated effect of the ecological divergence between parental populations does not result and associated with overall genetic divergence are the it doesn't sound like it's saying anything that you're saying, man.
You just missed a sentence.
I'm reading the whole thing. We'll go through it again.
No, you just missed a sentence.
Stop interrupting. Stop interrupting, please. I got it in front of me. I can read it again if you want me to. Just ask me to read it again. It says that test how they correlate with restrictions of genetic admixture upon secondary contact. Our results indicates that the isolated effect of ecological divergence between parental populations does not result in reproductive. I'm sorry, guys. I got my.
Reproductively isolated genetic transitions between parental.
Hold on. Isolated taxa, even the genetic transitions between parental taxa are narrow instead of the process associated with the overall genetic divergence are the best predictors of reproductive isolation. And when parental taxa diverge in nuclear markers, we observe the complete cessation of hybridization, even to sympatric occurrence of distinct evolutionary lineages. Although every parental population has diverged into mitochondrial DNA, its degree of divergence does not predict the external of RI. So what is your point?
It should be quite obvious.
Okay, so
the concluding section is like the results show that instantaneous the evolutionary outcomes of ecological divergence differ from those of genetic divergence. While evident properties of taxa may emerge via ecological divergence, such as adaptation to local environment, RI is likely to be a byproduct of processes that can contribute to the overall genetic divergence, such as time of geographic isolation, rather than being a direct outcome of local adaptation.
Okay, well, the point is that this is not saying that the genetic divergence is keeping them from, that's not saying anything about them not being able to interbreed anymore. Of course, there's going to be genetic divergence. That's going to happen, when species vary anyways to begin with. But how is this saying that the genetic divergence indicates that they cannot no longer interbreed? Because we would expect that anyways, right? We would expect genetic divergence we see genetic divergence with wolves and dogs and coyotes.
You'd go to the section on.
You said that this section.
Ecological divergence as a factor of reproductive isolation.
So this actually happened with humans. I don't see what the big deal is here.
I tell you what the big deal is. He said that he was telling me that genes. That they were separated by genes so that they can no longer interbreed. This doesn't say that. It's just talking about genetic divergence. That's all it's saying. We already know that, and you're going to see genetic divergence, even with, like I just mentioned, dogs and wolves. But they're not separated from being able to breed by that genetic divergence. So nothing that he's saying is connecting with what he was saying. There's not anything here saying that the genetic divergence is keeping them from interbreeding with each other. I think that would be obviously a silly thing to say, because we know that there are some species, like what we were talking about earlier, lions and tigers, they can be interbred.
Well, even a little less contentious one is like elephants, right? They talk to any elephantologist. What would the term be? There are 3 species of elephants because they tend to group and gather. They don't interbreed. Now, can you take an African, what are they called, the African bush elephant, the African forest elephant? Can you interbreed them? Probably. I guess so. I don't know. Who cares? Because that's not really where the species boundary is in whether or not it's possible to breed them. But biologists classify species by, well, there's that group of animals over there and that group of animals. I don't mean, when I say over there, I don't mean geographically. I mean when you talk about a population, you mean an interacting population, and in the case of biology, you mean reproducing. And the fact of the matter is that those 2 types of African elephants don't interbreed, so any biologist would classify them as separate species.
I've got another
Even though technically
prediction that I want to throw out to you guys.
Yeah, well, we touched on this earlier. I mean, I wouldn't say that, like, for example, like I brought up earlier with tigers and lions, that's some sort of speciation event. So I think that's like the main point right here. All I was doing by bringing in the tigers and the lions was that if we're observing the changes happening now, and we can see that, two species like that, or two, the lion and the tiger, that they can interbreed with each other, I think that obviously that would indicate that that's not necessarily true. I mean, like the finches, that they're not seeing breeding with each other anymore in the Galapagos Island. I believe it's Daphne major. They haven't been seen interbreeding with each other as well. There's a large divergence there, but that doesn't necessarily mean that they can't be interbred, because we know that we can take cheetahs and take their dirt, mix them with lions, and you can get a hybrid with that. So I mean, you can do the same thing just because they're not seen interbreeding with each other. That doesn't necessarily mean that they won't interbreed. I mean, and again, the article that he was giving me didn't say anything about the genetic divergence keeping them from being able to interbreed. It just talked about the genetic divergence, and it just didn't connect with what he was saying.
Can I take a step back? I want to go back to something that was said about the testability of evolution, and how we could actually. There are methods that it could actually be overturned. Now, I want to go back to the honesty of evolution, and I feel like that there was this idea that the perfect example is, and I mentioned it earlier, and I got some flack for it because I didn't really get a chance to explain it, but the perfect example is chromosome number 2. This was a testable hypothesis for evolution. And it actually presented the problem for evolution originally, because if apes and humans truly had a common ancestor, then we should have the same number of chromosomes, but we don't. And that was actually an issue for early evolutionists. Humans having only 23 pairs and apes having 24. And so this is a method to test the honesty, integrity of the theory of evolution, and evolution came out on top.
So I have a question.
Well, sorry, can I just do chromosome number 2 thing, because he mentioned that before, and I think before, that's right, Stephen.
Go ahead, Hogtie.
When you mentioned that, Eddie, before when you mentioned chromosome number 2 being a prediction, I thought what you meant was that evolution would predict those kind of fusions. And I was very quick, even interrupted you to disagree with that. I don't think evolution made the prediction specifically that, well, this fusion would happen, because keep in mind, by the time evolution theory came along, there had already been, the other apes with their 48 chromosomes, and humans with the 46. So you wouldn't say, because what I'm saying is that the observation of the numbers of chromosomes was made before evolution was really discussed as a theory. So you wouldn't say, well, so maybe I misunderstood you. I thought you were saying that evolution would predict the fusion. But given the
evolutionary biologists did predict that at some point a chromosome fusion happened. That was a prediction by evolutionary biologists.
Well, that's not a prediction.
I don't know how many sources to cite, but that was a prediction specifically made.
No, that's my point. That's not a prediction then, because the thing is, they already knew. Well, I'm sorry. What am I saying? What the hell? I cannot believe I just said that.
There was no confirmation of a fusion on the chromosomes. At this point, there had been no. We didn't have the means to test that.
That's right. Thank you. That was a terrible. That was a ridiculous thing. I said it was so bad. I said, evolution's been around. We already knew that they had this many chromosomes before evolution came around. That's ridiculous. We didn't know about chromosomes back then. I'm so sorry. That was ridiculous.
Prior to being able to test, and see, this goes back to, they asked for a specific demonstration of a predictive measure that couldn't be tested. They gave specific parameters. They said, well, it has to be something, they give an example of Mars. If they did something on Mars and we had no way to test it and find out what's true, that's a way to prove it. Well, this is what chromosome number 2 did.
What I'm saying is that evolution
specifically predicted.
Evolution wouldn't necessarily predict that specific fusion from happening. I'm saying that once they made the observation, like you think, well, wait a minute. So one of the features of apes is that they all have 48 chromosomes. oh, it turns out here's one type of purported ape that has 46. Well, that's really puzzling. Well, if evolution's true, that is, sorry, we keep saying evolution, we mean common ancestry. And in this case, we mean specifically common ancestry amongst the great apes, including humans, under the theory. That is a big kick in the teeth for evolutionary theory. So, but if it's true, then what we'd expect to find, now let's make a prediction about an observation we hadn't made yet, and that is about the telomeric sequences.
Yeah, and
that's a very risky prediction. Very risky prediction to say, well, if it's the case that chromosome number 2, given the size and shape of it, is actually a fusion of, and I forget which it is, 2 other chromosomes, well, then what we would expect to see is too many telomeric sequences, and specifically in the locations that would make up for for this chromosome matching up with that chromosome. So if that's what you mean, that like they made the prediction that we would observe these multiple telomeric sequences, that's a very risky prediction. And then, yeah, I would agree that is one that was made and confirmed.
Yes, it was. Because this whole, the difference between humans having 23 pairs and apes having 24 pairs was a serious problem, because at any point, if a chromosome was spontaneously lost or gained between offspring, it would result in serious consequences. It would end in either miscarriage or phenotypic disorder like Down syndrome or Turner syndrome. And this was a major problem. And evolutionary, and as you properly stated, this was a very risky proposition to propose that this was the case. And lo and behold, it is the case. To me, this is nail in the coffin on anybody who refuses to accept.
Yeah, the problem here's the problem with it. The chromosomes on the chromosome 2. Yeah, I'm sorry. Go ahead, man.
Okay, so this is it goes back to what I was talking about earlier. Let's just say one of the predictions that you just made, that you just brought up, right? Let's just say I use a completely different method to make that prediction, or to make a prediction that's close to that, right? And I label myself as an ID proponent. Would that put it under the ID category? Listen, science is science. Evolution, it's a theory, it's a scientific theory, but it's not science itself. Like, that's the whole point of it. The scientific method it's a tool that we use to make predictions and study natural phenomena, right? And that's all it is, right?
Well,
theory.
Science is the tool. But
the problem is, I want to respond real quick, guys. I'm sorry, because I've been trying to stay quiet for a while. Can I respond really quick? It wasn't a fusion event. I mean, it wasn't a fusion event. It's declared as it being a fusion event, but when you look at the chromosomes, what you're actually seeing is an absence of telomeric repeats, and you would expect to see that very clear distinction if the two chromosomes had fused together, but you don't see that. Well, let me put it this way: they're absent to the point to where you can't tell if they actually fused.
Right? I did mention a moment ago too. I said that I think there is rock solid evidence that that's a fusion because of there's just too many telomeric sequences for it to be one chromosome. Although I did mention sort of under my breath, and it's hard to tell what gets heard when multiple people talk, but I did say, but that point is under dispute. Now, to clarify, there's precisely one author I know of who's I believe he's an ID proponent, and Drew, you know who I'm thinking of? Do you remember the name? I think you'd even reshared some of his stuff a while ago. I don't think it was B.
Agax.
Is it London? I don't
remember. Well, let me just limit it to what I do know. Then is that this is the idea that chromosome 2 is a fusion has not gone unchallenged by the ID or creationist community, and there is actually a paper out now that shows that there's protein coding sequences that span, we would call it transtelomeric. So this internal telomere which doesn't make sense to have a telomere inside a chromosome. But there's the claim now is that there's a protein coding sequence that actually starts before the telomere, goes right through it, and finishes on the other side of the telomere. That makes no sense at all if it's a fusion. I mean, somebody would have to come up with a different theoretical account for that. But it turns out that that dispute is itself under dispute. There are people who know far more than I do about how sequencing works. That say that no, basically the way that this comes out is somebody sat down with because we have the whole human genome available online, right? And this guy sat down and looked at it and said, well, hey, when I look at this, I see this protein coding sequence that goes across here, and other people that know more about it, I don't know how he came to that conclusion, and other people who say know more about it than I do said, no, here's why the method he used was wrong. And to be honest with you, I don't know how he came up with it. I don't know what their refutation is. So I'm just suggesting to people in the room, keep your ears open. If you are sure that it's a fusion, be aware that it's something that's disputed by this particular author. And if you're a big fan of this author, keep in mind that there are people out there who've written fairly credible rebuttals to it. But unfortunately, I don't know enough about either.
There's been geneticists as well that have actually critiqued it, because you would expect if you had two chromosomes fusing, you would be able to see the patterns within the telomeric sequences all the way across the board. So you would be able it would be very distinctive. You'd be able to see that if it were two chromosomes that fuse, and you don't see that. I mean, just saying that because you got it's too large of sequences, that therefore it's too big. It cannot be just one. I don't think that's really a good answer, because I think that if a chromosome has a specific pattern to it, and you can tell. I mean, you can. Well, that's actually what's going to make it distinctive.
Well, how would you tell? You are talking about the telomeric sequence, right? When you say you could tell, how would you tell?
Well, what happens is that when you're looking at the chromosome, you can see the patterns. There's certain patterns that follow that chromosome. So I'm saying that if you have a chromosome attaching to it, and it's just a steady pattern all the way across the board, then you can't. There's no way for you to distinguish if it's actually two different chromosomes fusing to each other.
I don't know what you mean by a pattern. It doesn't go like AT all the way. Like what do you mean by a pattern?
Well, I'm talking about the telomeric patterns. I mean, you know what I mean by the telomeric patterns, right?
Well, I mean by pattern. I was calling it telomeric sequences. Like there's a sequence that's sort of famously a telomere. It's not a coding. It's saying, well, here's the end of the thing. Yeah, and in the case of chromosome 2, there's one at one end and one in the middle, and there's two in the middle, and then one at the other end.
Caps, yeah, you're talking about the caps on the end of the DNA, right?
Yeah.
Yeah, well, that's what I'm talking about. There's a specific patterns that you can see down the line on a chromosome. So if there were two chromosomes that fuse with each other, you would see that pattern to where it would be messed up. You would see a different pattern.
Different in what way? Again, do you accept what you do see is you see telomere and then coding sections, and then you see a centromere, and then more coding sections, and then you see telomere, and then more coding, and then another centromere that's inactive, and then more coding, and then another telomere.
Right. Yeah.
Is that not the pattern that's on chromosome 2?
Yeah, but obviously, if there were two chromosomes that fuse with each other, you'd be able to see the distinction in the pattern, right?
But that's what I'm saying. That's what they do see. That's what I don't understand.
Yeah, that is what they see. That's the whole point.
You see a steady flow of the cap, so you don't see that at all.
I don't see steady flow of the cap. What does that mean?
There's nothing at all indicating that it's two distinct.
No. There are four telomeres which begin and end in the chromosome, and two centromeres which are found at the center of the chromosome. I mean,
I don't think you're on what we're talking about.
Well, I'm sure I'm not. I just need you to explain what you mean by. Just explain it. So again, I just want to be clear. My understanding is that what they see is there is a telomere at each end like you'd normally expect, but in the middle there's a pair of telomeres, just one right after another. And then in between the one end and the pair of telomeres, there's a centromeric sequence. And the same on the other side. Yes. Right? Where normally in a chromosome you would see a telomere on each end and a centromere in the middle. This one has, telomere, centromere, telomere, telomere, centromere, telomere. It just looks like two chromosomes jammed together.
No, I think exactly what you would hope for if we shared common ancestry with with the apes.
No. What you would expect to see is you would expect to see a I don't know how to put it. You would expect to see something to alter or to disrupt the repeats that you would be seeing with the telomeric, what you're seeing with these different capies. That's what you would see. You'd see something that would be distinctive there. You wouldn't see a steady flow all the way across the board.
I don't know what that means, a steady flow all the way across the board, because you don't. You see these very distinct sequences.
You see steady, distinct sequences within one chromosome. That's the point.
But the sequences you see include a telomere, and then two more telomeres right side by side in the middle of it, and then another telomere on the end. Why else would that happen? Other than
because
fusion?
You're talking about you're talking about a fusion of chromosomes, and if you're seeing one chromosome, then you're not. I don't know what you're missing right here.
I don't either.
If I'm looking at one chromosome, right, and it's having a consistent pattern of these specific cappings, right, where are we going to be able to distinguish that there's a fusion event if you can't see that? If all you're seeing is one clean chromosome.
Well, if I look at
large, there's a lot there, but just because there's a lot there doesn't mean that there's two distinct chromosomes, and that's the point here. There's nothing.
No, you're missing the point.
Yeah, I think if I'm looking at
I'm not missing the point. I think I'm being pretty clear right here. I'm trying to understand where it is that you guys are thinking that there is a fusion event.
Okay, because if I'm looking at 23 chromosomes, so forget that it's 23 pairs, but let's just pick out one of each pair, because they're obviously comparable. The number one and the number one. So we pick out one of the 23 pairs, and 22 of them have this pattern where there are these long coding sections interspersed by other things. Let's not get into the epigenetics of it, but consistently you always see exactly two telomeric sequences, always one at one end and always one at the other end. And somewhere in between, there's a centromeric sequence, always one. But then I've got this one chromosome that's kind of on the big side, and it's got a telomere on the end and a telomere on the other end. But then in the middle, it's got two telomeres. In the middle of it, it's the only one that has in the middle of it. And then in between the pair of telomeres and the actual functional telomere, there's a centromere. And on the other side, between the pair of telomeres and the end cap on that end, there's a not functional centromere. I would say, well, how can that happen? And of course, if I have another blood sample from, an orangutan, and it's got 48 pairs of chromosomes, and it happens to be that number 2 is the same length as number whatever plus number whatever, I forget the numbers, I should look it up, light bulb might go on and say, well, here's a candidate explanation for it.
So I get perfectly.
Yeah.
No, because that's not exactly what you would see, right? You would expect it because you're looking at So if you're looking at the telomeres, what you would basically expect to see would be you would look at the stability of the loops, right, and the motifs. That's what you would try to look at. If I think you were to find something that was inside of the chromosome, you would try to look for something that would be outside of the patterns of that specific looping. Do you get what I'm trying to say? So if you're seeing...
Not even slightly. Let me ask you this. How do you think, so the proposed fusion, which I'll call it proposed fusion because I understand it's under dispute, not everyone accepts it, but under their view, how do you think the fusion took place? I mean, is it a side-by-side snuggling up or an end-to-end?
No, what I'm trying to say is that with the chromosome, you're seeing specific patterns inside of that chromosome, right? You're going to be seeing specific patterns in this chromosome. If you're seeing a steady pattern in one chromosome and the chromosomes fused at the end, then you would be seeing a different pattern, right? You would see where it would be disrupted. But we're not seeing that.
Yeah, okay, yes, you do. But the thing is, you see that in every chromosome, where if you were to lay out a chromosome on the table in front of you, which whatever that would mean, you see Like there's no. That's why I was confused by the word pattern, because it's not a repeating. That's not what you meant, but there's no pattern to a chromosome in that it's not like AT, AT, AT over and over again, right? There's different bands. There's different long stretches of it that'll code for this protein or that protein. And if you look at one end of a chromosome, you're going to find a telomeric sequence, and then something that encodes for whatever that end of the chromosome encodes for. And right after that, there's going to be another sequence that encodes for something else, and another sequence that encodes for something else. And then there's going to be a centromere, and then more, I don't know, whatever this is for. But the other end of the chromosome is going to code for something else. With chromosome 2, that's what you see. You don't see a consistent pattern across all of it, but you don't see a pattern across any chromosome. In its growth structure, the thing that's consistent pattern is always a telomere, one at each end, and a centromere, not in the exact middle, but somewhere between. And chromosome 2 is the one exception, because it's got a telomere, and then some stuff, and then a centromere and some stuff, and then a pair of telomeres, and then some stuff and another centromere, and then some stuff and a telomere. And that's exactly what you would expect if it was an end-to-end fusion of two different chromosomes.
Yeah, but that's the problem, is that we're not seeing anything like that with the chromosome, with the fusion of these two chromosomes, like what's being claimed.
That's exactly what we're seeing.
No, you're seeing an absence of those tandem repeats.
What do you mean tandem repeats? You're talking about the two telomeres side by side?
Yeah, I'm talking about the capping. You would be able to see right there at that end where they were fusing together. You would see a distinction where they had fused with each other, but we don't see that. You
do. That's what I'm saying. You see two chunks in the middle of chromosome 2, two chunks of non-coding telomeric sequences. They're just looking at the telomere.
Okay, but you do also see non-coding regions elsewhere, right?
Yeah, like.
So I mean, if you're seeing I'm sorry.
Yeah, true. Go ahead and click on that link I posted in the chat, the last link.
Okay, let's see this. Okay, you're showing me the fusion and at the fusion towards the end.
Show
right?
what we're talking about. I mean, I don't see how you. I mean, to me, it's like it just comes across as no matter what.
Oh no, but you see things like this throughout the chromosome, though. That's the point. This isn't like as straightforward as what you're trying to show me right now. What I'm trying to say is that the fusion event it wouldn't look like this. You wouldn't expect to see a consistent pattern of these cappings, right? So I mean, why are you showing me this with this right here?
Well, I'm trying to post that into the live chat, but I remember I can't post links in your. Can I post anything? Oh, this is your other channel. I can post. Okay, never mind. I don't know what to tell you about this, True, because this is a reliable observation. I mean, and Eddie just posted like sketches that are based on photographs, and the photograph is right beside it. And I swear I'm not understanding what your criticism is. Like you're saying that we're not seeing that, referring to the fusion location.
Well,
but we're saying yes, we are.
Yeah, because they exist because they're existing as independent monomers. That's what we're seeing.
They exist. What do you mean they exist as independent monomers?
Well, I'm saying that. Okay, so you're looking at the telomeric repeats that are only existing as monomers, but you're not seeing the tandem repeats that that would show a flow of two distinct chromosomes fusing with each other, right?
I think I don't know what you mean by tandem repeats or distinct monomers. Like I don't really know how.
Yeah, I don't really know how much clearer I can make it. I mean, if you're looking at a flow of two different chromosomes that are fusing, you would see a distinction of two chromosomes fusing. But the pattern of those chromosomes seem to be consistent all the way across the board, as if it's one chromosome.
I mean, I don't see what you don't see. That is what you are seeing. You're seeing a pattern of two chromosomes.
No, that type of pattern you're seeing in different places, though. So how do we know that that's the place where they were fused? I mean, the point is that what you're looking at, it seems like it's one chromosome. That's what it looks like.
Like in the photograph? Is that what you mean? Because you have to do sequencing to know what a telomere. Like, I mean, I don't know. Okay, look at the ends of the chromosome in the picture. On the let's call it the south end, the bottom end of the human chromosome 2, it's kind of grayish, and then it's kind of darker gray a little above that, and then there's some more bands. Go to the other end, well, it's kind of a dark gray. Like, you can't tell what a telomeric sequence is by looking at a photograph of it.
Yeah, but if it's in the independent monomers the way that it is, I mean, fusion events, they can take place like this, but usually whenever, like we're looking at with this chromosome, there's actually an effect that actually takes place within when chromosomes fuse like this, to where they actually self-destruct. So I mean, it wouldn't happen like this. The way that the independent monomers are set up in here, it looks like it's a steady one chromosome.
Well, in the photograph, yeah, but if you sequence it, there are like hallmark telomeric sequences. Every telomere of every chromosome in the human body has a recognizable telomeric sequence. I mean, I think you got to do some reading on.
Yeah, that's what I'm saying.
When I use the term T.
That's what I'm saying. We don't see a distinction of two chromosomes fusing right there. I mean, this event.
It's a picture.
What is the word for it? What is the word for it? What is the word? You're showing me the picture, but I don't think you're getting the picture, Eddie, because the picture that you're showing me is showing what I'm questioning right here. I mean, that we can't say that the fusion event happened at that point.
Well, what he's not showing you, he's showing you a picture of the actual photograph of the human chromosome, and I guess chimp, they called it 2A and 2B.
Now, what is it called? What is it called? I'm looking for the word. I don't know what the word is called. It's when this type of event happens with the monomers. I don't remember what it's called, but it actually causes it to actually self-destruct.
Can I ask a quick question?
Do you know what the word is that I'm looking for, Hogtie?
No. Like, are you talking about like when there's translocations?
You're trying to ask a question.
No. I don't remember what the word was. It's been such a long time since I've looked into this. It's like apoptosis or something like that.
Apoptosis.
Is that what I'm looking for?
Apoptosis is self-destruction, yes.
Apoptosis. I think that's exactly what would happen in this event.
Why do you think that?
What are we talking about?
Well, because usually when this type of event happens with the chromosomes, this is what would actually happen. It would self-destruct.
I got to pick you up on what you mean by usually. Like what experience do you rely on to know what usually happens when chromosomes fuse in species?
Perfect.
Well, what I'm saying is that, I mean, whenever this type of event happens, it's actually hazardous to the cell, so it self-destructs. There's a similar process that actually happens the same way with what is it called? It's the P57 gene, I believe it is. When there's a damaged cell inside of something damaged inside of the cell, it'll self-destruct to keep that cell from replicating.
Can I ask a question?
So there's a mechanism. There's a mechanism that's put in place that keeps the damaged cell from reproducing. That's what apoptosis basically does.
Okay.
So if we're looking at with the sequences that we're looking here, and we're looking at the monomers, and we're looking at the capping, and you're saying that it fused the way that it did, that's going to be hazardous to this because usually in other, like there was a case of somebody that actually his chromosomes actually fused together, I believe, but I don't remember how exactly it fused, but it didn't fuse together like this, and I like that. That's okay, but right here, it's the way that the motifs are actually being shown. This is actually like one straight chromosome.
The way
can I ask a question?
Just hang on a sec, Backminder. Just because the thing is, right now you're making a bunch of, and I'm going to say just wild, unfounded assertions. You're saying that, well, usually this would happen. This is what I would expect to happen. And
you know what apoptosis is, right? That's what apoptosis is.
Sure, but what you're doing
that's what happens when there's something going wrong with it. I mean, it's self-destruct.
Right, you're making a declaration that anytime there's an end-to-end chromosome fusion, it's going to lead to cell death. And you don't know that to be the case. What you're saying,
this is.
I'll distill.
No. This right here isn't an end-to-end chromosome fusion. That's what I'm trying to say about this.
I know that's what you're trying to say. That's what you're asserting, and you have no basis to assert it. What you're saying is because
no, I'm saying that it's not event-to-event fusion because the capping isn't showing like you're saying it. I'm making a very clear statement right here.
I know you're making a clear statement. You're saying that that's not an end-to-end fusion, but I'm saying that theoretically, people who study this will say that yes, that's an end-to-end fusion. This is
okay. Well, that's not true because there are geneticists that say something different. I mean, so that's not true.
And there are also
it would be true that it wouldn't be true. Hold on, it would be actually true that there are scientists that say that, but there are also geneticists. I should have rephrased that differently because there's geneticists that say otherwise.
Yeah, the thing is, you can point to things and say, well, this would be a really, really weird chromosomal event, and you know what? I know of some other really, really weird chromosomal events, and that leads to cell death. Or to genetic anomalies. Well, therefore, a chromosome 2 fusion would have led to cell death or to genetic anomalies. And that's not the case. I mean, yeah, you may be able to point to any other number of chromosomal...
No, you're misunderstanding what I'm saying. I'm talking about the tandem repeats that you're finding inside of these genes. They look like a steady flow of a chromosome. That's what they look like. They look like a steady flow of one chromosome. You're trying to say that these are two chromosomes, but that the pattern doesn't follow that. It doesn't follow that there are two distinct chromosomes. It just shows that one steady flow of one chromosome.
Okay, sorry. Let's go back to your tandem repeats. What are you looking for when you're talking about? You're saying there should be tandem repeats or there shouldn't be?
No, I'm talking about that you would see a distinction in tandem repeats. If you're seeing a steady flow of a pattern inside of a chromosome with, just like, separate or like, random places to where you can see the capping, well, then you can't sit over here and say that, well, here's the capping event. Because I don't think it follows.
You're not answering my question about the tandem repeats. You keep mentioning tandem repeats, and I said, in response to that, I don't know what you mean by that. Like I know what tandem repeats are.
It's actually very simple. I'm trying to figure out where it is that you're saying that the chromosome events happen. You're giving me an example right there, but that is found in different places as well. You're not seeing that. You can't tell me where the fusion event specifically happened.
Yeah. Okay. Exactly. I can tell you exactly where it is. Where there's.
But you can't. That's the problem.
Yes, I can.
You can't tell me where it happened if there's a steady flow.
I would be able to.
Can I say something?
I can't. Just 1 second. I can because you've got a telomeric sequence on the end, and then you've got some stuff, including a centromere and some more stuff, and then you've got a telomere and another telomere. It's in between those two internal telomeres is where the fusion happened.
Okay, and here's the problem. You see other tandem repeats throughout the chromosome. I don't know how you're not getting this, man.
Okay, well, because I keep asking you, what do the tandem repeats have to do with it? There may be other places in the chromosome where there are tandem repeats, but that's not the point. They're not tandem repeats of telomeric sequences. They're tandem repeats of other things like encoding or controlling sequences, but they're not telomeric sequences.
Yeah, I don't know how you're not getting this, man. I mean, if you're seeing a steady flow of specific patterns in a chromosome, you can't point out one pattern in that chromosome if it's found other places as well, because then that's not necessarily. You can't say that it's a chromosome event. Then I mean, I don't know how you're not understanding it.
Well, it's because
it looks like one steady chromosome with specific tandem repeats, but you're picking out one spot and you're saying.
Okay, where are you getting this thing when you say specific pattern of tandem repeats? Where are you getting that, and how is it relevant?
Let fact finder ask a question because I don't get what I'm not understanding.
All right. I don't know much. I'm learning a lot. I hope I'm learning a lot. But I think maybe could you guys point to a specific fusion event that is known? Probably a fusion event that scientists can reproduce now so that we can compare what a fusion event looks like to what is being purported here?
I don't know of any that scientists have repeated. Like you generally wouldn't do a study where you're like, well, we're going to start gluing chromosomes together. Also, keep in mind, I don't do like molecular biology or genetics. So it's not really my area. But is it going to be a race between me and MM for? Can you remember the example? Is it the guy in China or whatever that? Or am I mixing it up with the Robertsonian? There's somebody who has a different number of chromosomes.
Yeah, he was in China.
Daryl's here. Daryl's here. Yeah.
Yeah, I know what you're talking about. He's who's in China, I believe. But yeah, actually, I mean, I might be saying something wrong, Hogtie. That's probably why I'm not explaining myself good, because it's been a while since I've actually talked about this topic. But I'm pretty sure that the points that you're showing me to, that you would have to see, you would have to show a difference in the repeats of these cappings. And what you're pointing to, I think you can look at different places, so you can't point out where the fusion events happen. I'm willing to be corrected on that, but based on what I'm hearing, I mean, I don't know why you're not getting what I'm saying.
Well, I'm not getting what you're saying because you mentioned the tandem repeats, and I don't consider the two telomeric sequences in the middle to be tandem repeats. That's not what a tandem repeat is. And I'm wondering, and I sort of first I didn't know why.
What do you? Yeah. Well, I know what you're talking about, but yeah. Hold on, Robbie. I get what you're saying and what you're talking about. You know, where you're going to see specific tandem repeats, but I think that you're misunderstanding what I'm saying about, that capping towards the end, because that's what you're saying where they fuse at, right? They're fusing together. That's what that picture is supposed to show.
Mm-hmm.
All right, there's a few points on this that are kind of just, I guess we're going in circles on. First, the homologous chromosomes 2p and 2q. In the non-human grades can be shown right there when laid end-to-end to create identical banding structure as a human chromosome. That's what we're talking about. Furthermore, the next thing I would say probably is the remains of the sequence that the chromosome has on its ends, aka the telomere, is found in the middle of the human chromosome 2, where the ancestral chromosomes fused. If you can see that, I mean,
but that's the problem. You don't see that in the middle. What it looks like with the chromosome is one big chromosome.
No. Actually,
there are certain patterns.
Wait a minute. The detail of the region, the pre-telomeric sequence, the telomeric sequence, the reverse telomeric sequence, and the pre-telomeric sequence is exactly what you would expect from a fusion.
Yeah.
I don't see.
That's not what you would expect. That's actually not what you would expect. Give me a moment. I'm going to screen share a video for you guys, because I don't think I'm doing a good job explaining this. So I'm going to screen share a video for you guys so that you guys can watch it.
That's where you would expect to find if a fusion had occurred in the middle of the human chromosome.
No, that's not what you. That's not at all what you would expect at all. Give me 1 second here.
Maybe I can be some help after the video, probably.
Give me 1 second here, guys, because the patterns that you're seeing.
The centromere. Furthermore, the centromere of the human chromosome 2.
Why do you keep on interrupting? Can you stop? Hey, can you stop talking for a second, please? I'm going to show you guys a video right now.
Sorry, I thought you were pulling the video up. I mean, I was trying to finish my point. So.
Yeah, I know, but
I guess my last thing I would say on that is, at the place where you would expect on the human chromosome, we find the remnants of chimp 2p centromere. That's the point.
No. Take a listen. Well, you might find it right there, but so I wouldn't disagree with that. But what I am disagreeing with is those patterns. Maybe this guy will explain it better than me, though. Give me 1 second.
How can you disagree with the patterns? You have a pre-telomeric sequence,
Which is
a
that
telomeric sequence.
Yeah, you got to be quiet for a second.
I mean,
second. Because I'm explaining myself, but I don't think I'm doing a good job explaining it. And give me 1 second. Why isn't it screen sharing? Give me 1 second, guys. I'm sorry. All right, do you guys see what I'm screen sharing?
Yep.
Yes, you can see.
Are we going to watch the whole 18-minute video?
Yeah, you're going to watch the whole video. This is chromosome fusion. This is a geneticist who's talking about this, okay?
I mean, this is craziness. I mean,
be quiet.
Videos all of a sudden?
Yeah,
Okay,
we're going to
if
screen
that's
share
the case-
videos.
I mean, come on, this is crazy.
I'm going to hide you from the
Look,
broadcast, and you're going to shut up. That's what you're going to
Look,
do.
man, this is his room. Don't act
Yeah.
the fool up in his room, man.
No, and that's fine. That's fine. I was actually kind of hoping, True, that because I was actually going to head off to bed here shortly, so I was going to sort of let this one topic go, and then I was going to say my goodnights. But if we're going to do an 18-minute, and I have seen this video. This is actually the one I was talking about. The one. Actually, he was interviewed by Ian Juby also.
Well, I hope you guys stick around for a second because this guy will explain it much better than I can. I mean, this was like 4 or 5 years ago when I looked into this stuff, so I've forgotten a lot about this, and I know that I'm not doing a good job explaining it. So I want just stick around for a little bit if you can. I think that he's
okay. I can stick around for a few more minutes, but and also just to give the endorsement too. If you watch his is it Wazaloo channel, Ian Juby does an interview with him. That's a really clear description of this issue.
Is that a creationist or an evolutionist?
Ian Juby, man, it's Ian Juby. Just because he's Canadian, he's a famous creationist. Just because he's a Canadian, don't pretend you don't know him.
All right.
Famous.
All right. I'm gonna play it. All right, guys.
Those wacky Canadians.
Great apes have 24 pairs of chromosomes, but humans only have 23 pairs.
Wait. Stop. Hey, do you know who's talking? True? That's Ian Juby. That voice you just heard, that's Ian Juby.
Right. I'll check him out later.
Was descended from the great apes. It was claimed then that 2 chromosomes fused together into one at some time in the past as humans were evolving. It was believed that human chromosome number 2 was the resultant chromosome composed of 2 fused ape chromosomes. We discussed this claim previously on several shows, as well as Crevo Rant number 181, and how the chromosomal fusion argument is bust. More news has come out since the airing of these shows and our previous interview with Dr. Jeffrey Tompkins, who joins me again. Dr. Tompkins' PhD is in genetics with a master's in plant science and a bachelor's in agriculture education, and he was on faculty in the Department of Genetics and Biochemistry at Clemson University for a decade. He also ran his own genomics institute for 5 years. He is now a research scientist with the Institute for Creation Research, where he continues to publish various papers. Some of those papers we're going to discuss today. He joins me from his office in Dallas. Welcome to the show again, Dr. Tompkins.
Thank you. Good to be back.
Now let's start off. I want to backtrack to a paper you came out with in 2011 addressing this claim that humans have a fused chromosome, which allegedly supports evolutionary claims.
Right.
So let's go back in time for a minute, back to 2006, and revisit a lecture given by evolutionist Dr. Kenneth Miller at Case Western University. The lecture is entitled The Collapse of Intelligent Design. Now, it's interesting to note he very confidently claims that not only is there evidence that humans had 2 chromosomes fused, but that this is such powerful proof of evolution that he brought it up in the Kitzmiller-Dover trial. A court case which frankly wasn't a court case, but a 3-ring circus complete with a circus judge. A circuit judge. The segment of Miller's lecture where he discusses the chromosome fusion was linked to all over the internet. In fact, even today, I get comments from atheists and skeptics pointing me to that segment posted on YouTube under the provocative title, How to Shut Up Pesky Creationists. They hand me this video as if I haven't seen it before, as if it's absolute proof of evolution. I'd like to play a segment of that lecture so we can hear straight from the horse's mouth exactly what Miller said.
What must have happened is that 1 pair of chromosomes must have gotten fused. So we should be able to look at our genome and discover that 1 of our chromosomes resulted from the fusion of 2 primate chromosomes. So we should be able to look around our genome. And you know what? If we don't find it, evolution is wrong. We don't share a common ancestor.
Okay, so in 2011, you and Dr. Jerry Bergman came out with a paper showing that this alleged fusion site was small, highly degenerate, and really not much of a fusion signature. 2 years later, you published a second paper showing that the fusion site actually contained the genetic code for an important feature inside a highly expressed life-supporting gene. Now, we discussed this when you were on the show last, and I got a firestorm of emails and comments saying that you were wrong, and even Kevin Miller said you were wrong. So clearly you were wrong.
Okay, well, should we backtrack and
Sure.
and cover the details of that discovery? Okay. Well, the fusion site actually is a functional feature inside an RNA helicase long non-coding RNA gene. So this gene produces functional long non-coding RNAs of various sizes. Now this gene has 3 exons, or 3 coding regions. The other areas of the messenger RNAs are spliced out of the transcripts made from this gene. So you get short variants and you get long variants of this gene. So Ken Miller actually gave an interview to a reporter that works for, what was that magazine? I've got it here, Mother Jones.
Oh, yes, yes.
Magazine. Does that sound familiar?
Yes,
okay.
yes.
So the reporter was a guy named Mooney. So Mooney basically got the interview from Ken Miller. So Ken Miller says that the fusion site is actually 1,300 bases away from the gene.
Right.
So a friend of mine at the Discovery Institute said, Jeff, did you see this article? And I said, no, I didn't. He said, well, this is what Miller is saying. He said, you need to contact him and get this thing settled. He said, you know, Ken Miller never responds to me whenever I send him an email, but maybe he'll respond to you. So I sent Ken Miller an email, and he actually responded within an hour. Very quick. And he was very cordial. He gave a nice line. But the interesting thing was he obviously didn't read my paper, because if he would have read my paper in its entirety, he wouldn't have said the things that he said. So I basically had to explain my entire paper to him over a course of several emails. And then he finally realized that he had made a mistake and that I was right. And so he said in the last email, talking about the long transcript, not the short transcript, the long messenger RNA produced from this gene that has all 3 exons. He says, in this transcript, the fusion site is in the middle of the first exon, as you know. Well, actually, it's not in the middle of the first exon. It's in the middle of the first intron. But anyways,
True.
I was right.
I've seen this exact video before. He doesn't talk about any research or any findings at all. He talks about an email exchange with Ken Miller for like 15 minutes. And in here he makes the claim that Ken Miller had to admit that he was wrong and that this guy, Tompkins, was right. And Ken Miller, in response, has put up the email correspondence on his blog. You can go read it and find out that that's not the admission that he made. This video doesn't at all talk about the findings. At one point he makes the claim.
Yeah, it does. You got to listen to him and pay attention to what he just said right before.
Okay, but we missed the part where he goes on about the back and forth. Oh, I sent Ken Miller email, and then Ken Miller wrote back, so I had to write back because I know Ken Miller doesn't read, blah, blah, blah. Can we get to the point where he talks about the research?
Is anyone there?
Yeah, I'm sorry, guys. Somebody came in. Give me a second.
Ken actually took this piece from this email where Miller acknowledged I was right and published it in an article in The Blaze, which is an online media operation. And so people can actually get this article that Casey Luskin published.
Hey, I'm going to take it off screen share, right, guys? Because I got to take something out of my documents that I don't want you guys to see, right? So I'm just going to play the audio.
With Ken Miller's quote from the email admitting I was right. So basically, everything is exactly as I described it, but not as I'm describing it really, but it's because that's what the data says. You know, one thing that's interesting about the fusion site, when they first discovered it back in 1992, now when I talk about when they first discovered the DNA evidence for it, when they sequenced what's called a DNA clone from human chromosome 2 from that region, when they found these telomere repeats, even though they were degenerate, it was not what they were looking for. They say that in the paper, that when fusions occur in nature.
So you guys heard what he said right now. That was not what they were expecting, so that deals with what you guys were talking about, it being a prediction. So just keep that in mind.
In animals that actually survive these things and sometimes even reproduce afterwards, that these fusion events always involve something called satellite DNA. So you can have, actually the PowerPoint presentation I gave you has got a picture describing it. So you can have a chromosome break and have two regions of satellite DNA fused to each other. You can have a telomere fused to satellite DNA, but you never see telomeres fusing to each other in living animals. Sometimes you will get telomeres that will fuse to each other in cancer cell lines, which is, I think it's safe to say that's not a healthy
No, definitely not.
living tissue, but I have evolutionists always say this. Yeah, well, look at cancer cell lines. Telomeres fuse in there. Well, that's not a healthy living tissue indicative of a healthy organism. Cancer genomes are completely rearranged and completely haywire.
Right, definitely not onwards, upwards
Because
evolution.
No, they're not. They don't represent,
Right.
functional, normal.
See, that's what I was trying to get at too. Like this type of event, if it were to take place the way that you guys were saying that it would take place, this type of event would have actually been dangerous or hazardous to life. So just continue to keep listening.
Cells.
Apoptosis, usually it causes it to self-destruct.
Anyways, we never see these things in living animals. So when they discovered this degenerate telomere region, it was not what they were looking for. They were looking for something that had satellite DNA in it. So if the fusion, if the human chromosome fusion was real, it would be the first documented instance of a telomere-telomere fusion ever occurring in nature.
Right.
But obviously that's not what it is.
Right, because two halves of the gene aren't going to come together from two chromosomes.
Well, telomeres are designed. Actually, telomeres are highly functional machines at the end of your chromosome.
Right, right.
They contain a very complex set of proteins that are grouped together called the shelterin protein complex that sits at the end of your chromosome, and the DNA at the end of your chromosome actually folds back into itself to form a DNA quadruplex, and then the shelterin complex sits down governing all of that and encasing all of that. And it's not just a static complex, it's actually a functional set of active proteins that also interact with elements of the cell cycle and various pathways inside your cell. So telomeres are actually functional machines, entities at the ends of your chromosomes that are doing all sorts of important things to maintain genome stability and to keep your cells functioning properly.
Right, right.
So they're not just these bare ends hanging out there waiting to fuse.
Right.
So they're designed not to fuse, basically.
Yeah.
When you think about linear chromosomes compared to the circular chromosomes in bacteria or archaea,
right,
you have a logistical problem at the ends. You have an engineering problem that must be solved, and not really a problem, but
right.
You know, from our perspective of intelligent design. But if you were a human engineer, it would be a serious issue to have to deal with and to solve.
Right, okay. Let alone blind chance.
Right.
So pesky creationists didn't shut up about this. All right. So if we can back up just for a second, because so the viewers can understand what we're talking about here. You said the fusion site produces a non-encoding RNA.
Well, it doesn't produce this gene does not produce a protein. So there's over twice as many genes in the human genome as there are protein coding genes that produce RNAs. And these RNAs do all sorts of things. Some of them stay in the nucleus. Some of them go out into the cytoplasm. A very small percentage are even used as signaling molecules in what are called exosomes outside the cell. But a lot of these long non-coding RNAs function in the nucleus, and they modify DNA. They're called chromatin-modifying RNAs. And then a lot of them also combine with proteins to form these big complexes with proteins. The RNAs sort of act as backbones for these proteins to bind onto, and they also contain regions that bind to DNA as well in the genome. In fact, there was a paper a few years back where this particular secular research paper said that the genome is like an RNA machine. So really, it's an RNA world in the nucleus of a cell in the genome. And so this gene produces long, non-coding RNAs of various different types. It's alternatively spliced. It has dual promoters. It has a promoter in front of the gene. Promoters are like switches in genes that turn the genes off and on and are involved in causing these genes to produce transcripts or RNAs at certain levels and certain amounts at certain rates in the right tissues at the right time. And so it turns out that the second promoter in this particular gene, it's called the DDX11L2 gene. The second promoter in this gene is the fusion site. So this is not just DNA sitting there doing nothing. The fusion site, and I have to use the term fusion site because it's really not a fusion site. It's a promoter inside a gene. And it's very common for genes to have promoters in their first intron inside them, as well as promoters in front of the gene. This is common across the human genome. And another important thing to understand is that you have genes on both strands of the DNA molecule. DNA is double-stranded. You have a plus strand and a minus strand. When we annotate genomes, we always annotate genes as to whether they're on the plus strand or the minus strand. And the genes run in opposite directions from each other on these strands. This particular gene is on the minus strand. So the message is not your typical message that's portrayed in the fusion site sequence. It's actually in the reverse complement of that sequence. So that's what the machinery in the nucleus, the various proteins that are called transcription factors are recognizing is in this other orientation on the minus strand. And so my new research that I have developed totally confirms that this fusion site is totally functional. It binds all sorts of transcription factors, over 50 different transcription factors, including RNA polymerase II, which is the main polymerase that transcribes genes in the genome. Okay, RNA polymerase II transcribes all the protein-coding genes and the long non-coding RNA genes. And in fact, these genes, these protein-coding genes and long non-coding RNA genes share many of the same transcription factors. But what's really cool about this gene that contains this alleged fusion site that functions as a second promoter is that it's highly networked with many other genes that are involved in high-level cell processes, high-level, very important cell processes. And so it's heavily networked with these other genes. It's heavily integrated into the genomic networks of the cell. It functions in over 250 different types of cell types that have been analyzed so far. And what's also really interesting is that my new research, mining the new ENCODE databases and this new version of a database in Japan called the Phantom database, which is also a database that studies gene expression, shows that this fusion site region, which is about 798 bases long, contains multiple start points for transcription where RNA polymerase II binds. So there's multiple transcription start points within this region, or transcription start sites. They call them TSSs for transcription start sites. So this is a hotbed of activity. It's not a fusion site. It's a functional region inside a gene that is completely